Well, good morning, good afternoon, everybody. Welcome in from across the continent for another exciting Exploring by the Seat of Your Pants broadcast. If you're joining us for the first time, so many of you are today, we are all about bringing conservation, adventure, and science into classrooms around the world. And we do this through about 30 live, free, interactive broadcasts with the coolest scientists and explorers on planet Earth every single month. Everything we do goes to our YouTube channel. So if you want to share this one, family, friends, 3 weeks, 3 years down the road, you can do that right there. Lots of opportunity to explore and discover some really, really cool stuff. Now, every now and then, Hannah from the Schmidt Ocean Institute reaches out and says, "Hey, we're live on a ship in the middle of the ocean somewhere. Would you want to come on and do a broadcast?" And the answer is always yes, because how often do you get to be on the aft deck of a ship in the middle of the ocean, sending robots down to the seafloor to explore cool things with awesome scientists? It's not every day. So it's very exciting to get to go back on the RV Falkor 2 to learn what their amazing science team is doing. A big thank you to all of you as an audience for being here today. And without further ado, I'm going to turn you over to our science team on the Falkor 2. Hello, everybody, and take us away.
Hello, can you hear us?
We can hear you fine. You're perfect.
Sweet. Thank you so much, Jesse, for that awesome intro. My name is Sophia. I'm the multimedia technician. I'm currently behind the camera, but we're super excited to bring you this ship-to-shore connection today with Explore by the Seat of Your Pants. Thank you so much. And I have these two wonderful special guests with me here today, our co-hosts that will help us lead through this connection. So when you have a moment, take it away and introduce yourselves and where we are.
Hi, I'm Claudia. I'm a graduate student at the University of Washington in Seattle, Washington. And today we're here on the aft deck of the Falkor 2, which is Schmidt Ocean Institute's ship. And behind us is something super interesting going on that we'll talk about in a minute.
Yes, so hi everyone. My name is Sydney Wilkinson. I am a postdoc at the University of Miami in Florida, so I'm really excited to tune in with you guys here in the middle of the ocean. I think our goal here is to kind of give you a tour of everything that we are up to and the project that we're interested in. So what we're really focused on here is looking at animals and the role of gut microbiomes in animals. So we're interested in animals that undergo diel migration, so they migrate from lower depths where they hide from predators in the daylight, and then migrate up to upper depths of the ocean during nighttime, where they can then feed on the nutrient-rich waters. And we're interested in understanding kind of how that migration incorporates and influences carbon flow in the ocean. So we study a lot of different things like phytoplankton and fishes and zooplankton in the ocean, and also their fecal pellets, so their poops that really contribute to the ocean.
Yeah, and something super interesting about this project is every level of the food web matters here. We're looking at the very base of the food web, which is our marine heterotrophic bacteria that are kind of generating these nutrients that phytoplankton and zooplankton then feed on during the daytime. And as they migrate down, that's where predators are able to eat them. The predators also feed on them when they're at the surface, but all of this food web is acting during the day and especially during the nighttime. And then the particles that are released from this cycle of the food web are also really important because we want to see what is kind of being locked down in the deeper layers of the ocean.
Yeah, so one question you might have is, how do you collect these animals, specifically fishes and invertebrates in the water? So we do so multiple ways, but currently being deployed back on the A-frame there is a giant mock nest. That stands for a multiple opening and closing nets with environmental sensing systems also attached to it. And basically it has multiple nets attached to a giant frame that's like 10 meters long. And you can send it down to different depths and kind of signal when you want the nets to be open to then trawl and collect those animals that are in the mid-water. So that's currently happening, which is really exciting. I think we're gonna retrieve it around 3 p.m. and we'll see lots of exciting animals to then sort.
Yeah, the cool part about the mock nest too is how it's able to sequentially fish what we call the water column. So like the entire span of water that exists that has different animals in each layer, it can capture different depth ranges essentially. So we're able to see these animals as they're coexisting with each other and as this food web is really in action, which is super cool. Yeah.
Super cool. Quick query. How many of these do you do every day? Is it one big sort of collection a day? Is it multiple times that you're doing this? Or is it like a set, range throughout one day and then the next day you go a little deeper or shallower? What's the deal?
That's a great question. So the net itself takes time to both deploy off of the deck and return onto the deck, sorry. And then it fishes for about 8 hours. And all of the nets are being deployed for different amounts of time. And then processing the net. So you process the collection tube of each net independently because they each fished different depth ranges. And that takes around 8 to 12 hours. So we can do multiple of these in a day, but it really is not ideal because sleep is also very important. And it's good to be thorough and take our time when we're collecting such precious animals.
Yeah, right now it's a lot of work. So we have kind of like 3 super stations kind of set for our cruise track. And in these super stations, we're kind of staying in the same area of the ocean. And we deploy the mock nests twice for night trawls and twice for day trawls. And then we move along our cruise track, kind of getting different eco provinces kind of along the way, along the coast. But yeah.
Super.
Perfect. Yeah, that's good context.
Yeah. Have you done this sort of expedition anywhere else? Is this the first time you've ever done anything quite like this? And are you planning on doing more? Like, is this just specific to this region, or is there more data to be gained pretty much anywhere with this exact same sort of thing?
So this kind of net specifically is, the shorthand for it is a Mach 10, which is the Mach nest net, 10 meters in length. And these are deployed in other regions throughout the world. Other groups do deploy them, but what's great about this cruise specifically is we're deploying the Mach nest net alongside a bunch of other kinds of sampling as well. So we're pairing it together, I think, for potentially the first time. And this region, I don't know that I've never done a cruise like this here. I've done a few very short, small cruises in the waters surrounding Washington State, but aside from that, no. And I'm excited to be here.
Yeah, I would just echo that. I think my experience is doing some short cruises in Alaska where I was previously, but yeah, this is the first time for myself to doing it here. I think there are plans for the bioreactors team to do another cruise in 2027 along the coast of Africa, I believe, but that is still in development. But yeah, it's all really new and exciting.
How long is the expedition, and how long are you going to be on it? Are you on the entire time that it's on the water? Do you trade out every now and then, or what's the deal?
We are out here for the full duration of the cruise. So it started, I don't remember the exact date.
Yeah, we were at port for a few days, but I think January 25th-ish. 23rd. All right. Yeah.
And we'll go until February 21st or 22nd, I believe.
Super cool. I also love that you checked your watch to figure out which date you came on the ship. Like me that does that.
The days go by here quite quickly. These are seawater incubators that, well, actually they're filled with fresh water, but we incubate seawater in these. They're temperature controlled. And this is to identify nutrient amendment that occurs from small microbial or phytoplankton based systems, but they can be used for all sorts of things as well. And they look really crazy, but it's a way to incubate things and keep them in a fairly similar environment to where we took them from. So these are similar to around 5 meters of depth, given the sunlight and the tent on here and the temperature.
Sweet. I think now it's a good time to head to the ROV hangar. And while we walk over there, Jesse would be happy to answer some more questions.
Absolutely. So first of all, the fact that we're going to an ROV hangar is super cool. So thank us for taking us there. You guys want to share in the chat? YouTubers, please feel free to dive in at any point. One of my big questions is, so you get these specimens, you're collecting things from all sorts of things in the water column. Can you process them and understand them on the ship, or do you need to take them back to labs in the US or elsewhere to get a real handle on what you found?
So a little bit of both. Our team is built or comprised of some experts in taxonomy at all different levels of like copepods, crustaceans, and fishes. Mainly based from, I think, Scripps and Chile. So we have experts on the boat that are able to like look at taxonomic keys and identify the species like on the boat. But that takes a lot of time. So sometimes if we run out of time or they're unsure of what species it may be, they save it in formalin and send it back to the lab and then do further processing and identification.
And that's just one type of the sampling that we do with the animals that we get from the big net. A lot of the other kinds, we do a bit of dissection here, or we keep the samples whole and we'll ship them back. And then there's a bunch of different processes that go on when we're back in our labs at home, because a lot of our interest in this specific cruise is looking at how animal guts transform organic matter in the ocean, how they eat things, and the bacteria in their gut are, we believe, to be really, really good at processing that material into usable compounds for other things or into little particles of carbon that get sequestered down into the deep. But we have no idea what the bacteria are specifically doing on a biochemical level, on a metabolic level for these organisms that they live within. So processing the samples in that way does have to wait until we're back home, but all of the collection and a lot of the taxonomic identification and sorting does happen here.
Very cool.
To the best of our ability.
Yeah. Thanks, ladies. If you're in where you need to be, you can take us a little journey there, and we'll take some more questions in a minute. This looks cool.
Okay.
All right, so right now we're at the ROV hangar. This is the beautiful ROV Sebastian.
This Washington license plate says "I break for science." It's a joke with ROV dives that when you have scientists there watching, they're always like, "Wait, we want to look at that," which is exactly what's been happening for us, meaning that the ROV has been breaking for science, and we've been using the ROV to collect animals that we see kind of in this mid-water region of the water column that we're interested in. And we'll walk around to the front in a second and you can see what are called jelly jars that essentially close around animals and collect them and bring them back up to the surface.
Yeah, I'm currently showing an overview clip of the ROV getting launched just so folks get an idea of the size and scale of the robot.
And it's really cool that we have the ROV as an option to like collect animals in the midwater, because when we collect them, they're able to stay like pretty protected in the jelly jars, and then they come up to the surface well maintained. As you can imagine, when you collect animals in trawls, sometimes they get a little bit deteriorated just from being trawled for so long. So it's really cool that we can get like these beautifully preserved specimens collected in this way.
Yeah, another cool thing that the ROV is able to do is when we do collect these specimens, it also has these bottles that are called Niskin bottles up here. There's 6 of them, and these essentially capture water right near where we've sampled, and so we know that this water is animal rich or has been around animals that we've just sampled, which is great because then we can look for specific nutrients or animal-derived biomolecules within these samples, and we have confirmation that samples that we're interested in, the animal samples that we're interested in, were just there, which is cool. And then these are the jelly jars. They're quite large, and so this section, and there's also a section just like this slide over the top when they're collecting an organism that we want to bring back up.
Super, super cool. A question from our Monty Tech crew, grade 11s. They want to know, is there one species or one family you're particularly focusing on, or is this just the full breadth of everything that you can collect because it's all very interesting?
Yeah, we're trying to get kind of a full breadth to kind of see the diversity that's happening in the midwater, but we are really interested in the dominant species that are present, assuming that they probably contribute a lot to like organic matter flow and to particulate organic matters that get exported into the ocean. So kind of a little bit of both. And a lot of the times we're not sure what we will kind of encounter. So it's all exciting for us kind of at the end of each trawl or when the ROV is deployed.
And sometimes it's good to, sometimes we're thinking more about collecting functional groups instead of specific species. So things that migrate, things that stay in the same sort of layers, things that play a specific like ecological role. We're also trying to consider when we're sampling.
Yeah. Quick note, because those are huge specimen jars, and like you're doing multiple dives. Where are you storing all these? Like, how many are you trying to collect over your month-long expedition? It seems like half the ship would end up with specimens by the time you're done.
We'll definitely have to show you the freezers that we have that are almost, they're getting fuller and fuller, and we've only done, I think, like 3 trawls now. So we're expecting very full freezers by the end of this cruise.
Yeah.
Very, very cool. If there's anything else you want to walk us around and show us, by all means, we can do a bit of a journey. We can check out those freezers or start heading in that general direction. We've got more questions coming in all the time, so we could really take this as long as you would like, but it's very exciting. Is there a personal favorite creature that you've both found so far, given that this is one of your first expeditions or first one in this area, like sort of go-to coolest animal?
I think the last trawl, it was super cool. We saw like a female lanternfish. I should have sent a photo to show via Sophia, but that was the first time I saw a lanternfish like in the flesh. I've only seen them like in photos before, so that was really cool for me.
That and whalefish too. That was super cool. Honestly, everything. It's such a joy, like, to see it. Yeah.
By the way, your reactions are exactly what I like to highlight to students, because I think a lot of people assume that scientists are very serious professionals. And in fact, we're all just super nerds and we just love all this stuff. And it's so cool to see it in person. So everyone should check out a lanternfish. That's my favorite. And I can't encourage you to go look that up enough because they're magical, wonderful creatures. With that, I'm going to take a question live from our class, actually. So I'm going to bring in our Monty Tech crew for another one. And then we'll keep going. We'll walk around some more, take some from our YouTube friends. But Monty Tech, if you want to come on in, you're good to go. Hey.
I was wondering if you guys
So the good news is you like to be on camera, which is awesome. The bad news is it's really hard to hear you. So if you can repeat that a little closer, that would be lovely.
Can you hear me?
A little bit better. Yeah, try again.
He's asking, do you guys keep the organisms, or do you then, like, once you study them and catalog them,
Yeah.
put them back into the water?
Are you putting them back in the water at the end? Are you keeping them? Do they even survive the process of collecting them? So all good details. Perfect question.
The majority of animals, unfortunately, do not survive the trawling process, but we do try to do quantitative sampling, so we take everything and we store it in some way.
Yeah, and we actually do need the physical samples to do kind of the techniques Claudia mentioned earlier back in our home labs. So we need different tissue types and stuff to kind of look at their isotopic signatures or their metabolites, like whatever we're interested in, we do need the physical tissue, which means that, yeah, we do keep a lot of the specimens that we collect.
I want to stress, because it's a really good question to have, in particular to younger audiences sort of feel weird that we do sometimes have to take animals and that they die when we capture them. It's such a small proportion of the things that are taken from the ocean and the world, and it does go towards understanding those species and those habitats so much more. So sometimes it actually is important for science for things to die, which sounds a little weird, but I'm really, really glad when we get this question, and thank you both for answering it. Miss Grimm's class in Tucson wants a follow-up question, which is, do you ever bring up sunken trash? Do you catch things that you don't want to catch? Are you reading through plastic to get the creatures that you want when you're doing these trawls?
I've yet to see trash or plastic in the actual trawl material, but we are pretty remote out here, and we have seen like some floating plastic water bottles just on the surface.
And I will say I do a lot of filtering of like animal or like waters that have been incubated with animals. And on the filter, you can see kind of small like microplastic threads. So they're very small, but like not like the larger plastic chunks you might like imagine. But I have noticed like small little bits.
Yeah. Hey, comforting to know that it's. Oh, sorry. Go ahead, Sophia.
I was going to say, on previous expeditions, at least that I've been on, oftentimes in the control room, we do end up collecting plastic that we see on the bottom of the ocean floor if there aren't too many animals living on it, because sometimes animals do find their way onto pieces of anthropogenic waste. So, yeah, sorry, I just wanted to hop in there real quick.
Oh, that's fantastic. No, I think it's really comforting to know that it's not mostly plastic that you're picking up. And certainly we've covered a lot of expeditions here at Exploring by the Seat of Your Pants, and plastic is everywhere. It's on the top of Mount Everest. It's in the bottom of the ocean. We do find it wherever we look. But comforting that most of your stuff is animals so far. It seemed like you were going to explore something else, and I might have interrupted you earlier. So if there is anywhere that you wanted to walk, by all means do that. We've still got more questions pouring in, or I could keep going with Q&A, whatever you folks want.
I was going to say, we're going to continue on to the CTD and to other areas of our science spaces, but while we're heading over across to the other side of the hangar, maybe we can do one more question.
Okay. Sure. So this one might be for you, Sophia. I think you know most offhand, but Xenon wants to know, how long did the ship take to construct?
Ooh, that's a really good question. So this vessel was actually refit from an industry vessel that was formerly called MV Polar Queen. And so that one was built in like 2010, I believe, like early 2010s. And it was refit starting in 2020 in Vigo, Spain. And I started working here in 2022 at the end of that refit period. So we actually worked really hard in a shipyard to convert all of the industry spaces into science spaces. I hope that answers your question.
That's fantastic. Yes, and I can pass along the full Falkor 2 website to everyone on the YouTube chat and StreamYard chat, so you can check out the full construction. There's all sorts of great stuff on the Schmidt Ocean Institute website as well. Cool, guys. All right, what's this wicked cool science-looking thing behind you two?
So I know Claudia pointed out the Niskin bottles on the ROV. This CTD here is very dominant in Niskin bottles. So we use this to collect water at different depths. And it's also fitted with different sensors as well to kind of get like environmental parameters. Claudia, I know you sample from this, so maybe you can chat a little bit about what you're interested from the water that we sample.
Yeah, totally. So just like Sydney said, while the ROV had 6 Niskins, this has 24, and each of these holds 12 liters of water versus those only hold 5 liters. So a ton of water can be collected with this, and the CTD, this is called a CTD rosette. The rosette just meaning that all of these are circular, and the CTD actually stands for conductivity, temperature, and depth, which are 3 parameters that are like very, very core to oceanography. Basically, it's hard to imagine, and it looks crazy, but this entire door opens and swings out, and then the CTD follows on this cable, and we deploy it over the side, and it goes down. It can reach all the way down to the bottom of the ocean, which here is around 6,000 meters. Crazy, but we have been deploying it slightly shallower, and essentially all of these bottles actually go down open, and then when they're at the depth that we want, we close them, and that seals away the water to be brought up for us to sample from. And CTDs are used primarily to sample, well, actually so many things, but a lot of nutrient work, a lot of like basic ocean chemistry parameters can be measured from the water that we get from a CTD. The list goes on. Like, you can do so much from this water, and it's a great resource. And it's really amazing that this one has 24 Niskins, because each time that it goes out is a whole endeavor, and a lot of bottles have to be prepped, and a lot of lines have to be washed, and people have to be around to process the water when it comes up. So this is also a big sampling effort as well. Yeah.
Quick query, and this might be totally outside of the scope of the research you do with that, but can you or do you collect any eDNA when you're getting those samples? And this is just like DNA in the water, so you can understand the species that happen to live in those areas, or not?
Yes, eDNA is collected from a CTD occasionally. Unfortunately, you have to sample quite a bit of water to get a quality eDNA read, from my understanding. So each of these holds about 12 liters, and depending on what depth you go to, you need to filter around 20 liters of water minimum to get one replicate of an eDNA sample. So it's it's quite a lot of water, but you absolutely can.
And as you can imagine, there's a lot of different scientists interested in the water. So there's kind of a water budget we have.
Oh, why don't we show it?
So people have designated Niskin bottles that they're interested in. So it requires a lot of planning and sharing of water and collaboration. But yeah, not all of this water is for like one thing per se.
Yeah, so on here you can see like this is the bottle number, this is the depth, the group, and it goes on. And so this bottle number 1 on the CTD went to 1,000 meters, and the group sampling for it are the OBV group, which is kind of like our core measurement team, and then WAM, which is another group that is here on this cruise with us. And so each bottle gets allocated to different people for different amounts, for different samples to be collected. It's
a lot.
Very involved.
Yeah.
Well, a great segue from this. So our Monty Tech crew, and this is one of the joys of being in broadcasting, is that when you reach over grade 8, basically we get this question every time we have a broadcast, but just how much does this expedition cost and who's funding all this? Because it seems like a lot of cool science work. A lot of you are on board. Got some really cool robots and toys. What's going on?
I don't quite know all of how it works. My understanding is that, not including salaries, it's about $50,000 a day to run the ship and all of the operations. And then the group paying for this is Schmidt Science,
Yeah.
I believe. You're probably the best person to answer this.
I was going to say, we're basically a platform internationally for various visiting scientists for expeditions such as this one. And so we're funded philanthropically via the Schmidt family. And so Schmidt Ocean Institute is one entity. These guys are helped out by Schmidt Sciences, which is a sister entity, all funded through the Schmidt Family Foundation. So Schmidt Ocean Institute, excuse me. I'm like mixing the two. No, they're that close. Schmidt Ocean Institute is a nonprofit organization that is privately funded, and we aim to help scientists get their work done on this ship. And get
So I want to stress too, just for our audience sake, who might not know the context of this, but like I can literally think of no better privately funded science expedition platform in the world than what you guys do. Like I get to work with a lot of organizations, governments, agencies around the world, and like nobody brings in more scientists from around the world to do really cutting edge research in more places than Schmidt. So kudos to you for doing this. And I love this question. Like it's really important for us as a society to sort of question how we're funding this, why we're funding things like this. But really, there's some incredible work being done in the ocean right now, all around the globe. Schmidt's leading the charge in that. And it's just a really exciting time to be a scientist like our two amazing ladies today walking us around, because there's so much to explore and discover, and we're just starting to do it in the ocean in a really serious way. So thanks for the query and thanks for the answer, everybody.
Yeah, it means a lot coming from you, Jesse. All right, we're going to continue on and show some lab spaces.
Thank you.
The best space.
All right.
So first of all, I guess I'll just showcase all of the rain gear that's hanging because it's so prominent. We have beautiful weather today. Like the ocean is so glossy, it feels like a lake. But some days it can get a little bit rougher and we can have some swells. And as you can imagine, when you're pulling up nets, the nets are wet and you can get like completely soaked from that. So we have lots of rain gear to help with like the actual sampling. And then over here,
this is my lab group space. And these are some of all of the various kinds of bottles that we use to sample primarily from the CTD from. So it is quite hectic. But basically something that we do a lot of is filtering seawater. These are actually filter holders for, this is called a peristaltic pump, and it uses mechanical force to essentially force water through a filter that would be placed there. Another kind of filter holder is a vertical filter holder like this, where the filter, same as with these peristaltic pumps, gets placed in there, and then you pour water through it and a vacuum pump filters it out. And we have another vacuum pump set up over here for both small filters that would be placed straight through there, and these are larger filters where the filter gets placed on there. And you can see that it's kind of dark in this corner, and that's because we sample and try to measure vitamins in this corner of our lab, and vitamins are photosensitive. So typically we actually have the lights in here off so that we preserve our vitamin samples as best as we can and get a full measurement. But yes, it's quite crazy in here, and this is one of our freezers that I can open very quickly. I don't know how full it is, but this is some of the water that we get. So this is sampling for essentially dissolved nutrients, which is super cool. You can measure basically what's in the seawater itself, has kind of a background signal, which is awesome. Yeah.
Sweet. Is there anything currently in the cold lab?
I think they're prepping setup for the mock nest retrieval that's happening at 3 p.m. But this is our cold room. It's really handy to have because, as you can imagine, the animals that are coming up from depth are used to these cold temperatures. So to bring them onto like a warm boat would not be ideal for them. So first, the animals get put into this cold room. Each cod end from the mock nest gets put into the respective bucket for sorting, and then sorting happens kind of one cod end at a time just to ensure that the different levels in which they were sampled don't get mixed. So this room is very cold, but it's really good for the initial start of sorting. And then when it gets moved into the other lab for like taxonomic classification, all of the samples are put on ice to be kept cold.
Another
Cool.
thing that's in here is all of these liquid nitrogen storage containers.
Here, Claudia, could you come move a little bit this way? Your audio is coming out.
Yeah. All of these liquid nitrogen storage containers also hold samples that we'll be taking from this cruise. So we have our freezers and our fridges, and we also have all of these. So
Super, super cool. Here, we'll let you get a little bit warmer than the 4-degree temperature as we take a few quick questions. So one of them, just to confirm something, because we had this in the chat, when you're saying a certain phrase, mock nest, Loch Ness, one of our classes wants to know, how do you spell what you're saying?
Write it. Let's write it. But it's MOCNESS, and then again that stands for multiple opening closing nets environmental system or sensing system. So it's a huge acronym essentially. Yeah. But here's the spelling there.
There we are. Perfect. Thank you very much. So we had a cool question, which we get every time we go live on a ship in the middle of the ocean, which is, neither of you look like you're suffering from scurvy. And I want to stress that when I was a boy and I thought about expeditions, I was like, what are they eating on the ship? Can you tell us how well you're eating and what the setup is in the Falkor 2?
It's amazing. The chefs here are amazing. The food is great. We have breakfast, lunch, and dinner. And I feel like there's a wide range of available options, and they also include different like dietary restrictions, like if you're gluten-free or dairy-free. I think for lunch we had French fries and a chicken burrito and like spicy pumpkin soup, and there's so many salad options, so we're not suffering from scurvy. Yeah,
tons of fresh fruit still. It's delicious. Yeah,
I don't know if that is legitimately the chef that's on right now, but like, you know that a chef is good when they got like a full tattoo sleeve. Like, that's a good sign. Should be smoking on the back deck, sort of thing. Yeah, I'm
just showing them a picture of Chef Carl.
Oh, awesome.
For our audience's sake, I love when we get this question, because if you are a scientist in the future and you decide you want to go on expedition, if you do a land expedition, you're eating out of a plastic bag and you're pouring boiling water into it. If you do an ocean expedition, you got like a 5-star chef and it's the greatest food you've ever had in your life. That's pretty much a universal in our experience. So I'm really glad. Thank you for sharing all that.
Yeah. And
we've got one
to the main lab.
Yes, of course, the main lab. As we're going, can I ask you a question as we go?
Absolutely.
Great. I very specifically used a phrase earlier that I wanted to prompt a class to ask this question, which is, what on earth is eDNA? And it's the coolest thing ever, and we'd love to hear what you can tell us about it.
Essentially, eDNA stands for environmental DNA, and I don't study eDNA, so this is my best. But basically, eDNA, you capture an environmental sample, and from there you do basic genetic sequencing, or quite extensive genetic sequencing, to try and identify all of the bits and pieces of environmental DNA that exists within that sample from all possible inputs. So every organism that comes through that region that you've sampled likely leaves behind a little snippet of their genetic data, and you have to take quite a bit of, like for us, seawater, but you have to take quite a bit of that to essentially concentrate enough genetic material to be able to sequence it. And from there, you can kind of get a snapshot of sort of all of the big organisms that have been within that region. It's super cool and really amazing.
Like, as you can imagine, like as humans, we're kind of shedding bits of our skin. So if we were to collect kind of everybody's, like the air in this room, we would kind of get an idea based on the eDNA who was present, based on like what we are naturally shedding. So that you can kind of apply that to the ocean and what like organisms are kind of shedding in a way. Yeah.
What a creepy and wonderful analogy. Thank you so much. And I hope you find out who's been in the lab. I appreciate it. Tell us more about the main lab. This looks amazing. We've got about 5-6 more minutes together, so we're going to hang out for a bit, maybe take a few more questions, but any big things you want to highlight, by all means.
Yeah, it seems like the lab is pretty busy right now. There's a lot of scientists prepping for the mock nest retrieval. And also, I think some also sampling from the CTD that was run earlier today. I don't know if anybody, I think they're all really focused, so I'm not sure if they wanted to quickly say hi, but yeah, as you can imagine, this station gets very busy when we're about to retrieve the mock nests. Back here, this is kind of the sorting station. So this station here, Elon sorts through crustaceans.
Here's fish over here too.
Yes, there's like that fish station with a bunch of beautiful keys to kind of confirm what species are present. And then as they move through the stations here, this is kind of the measurement station where they have calipers and they get different lengths of the animals, and then also the weights of them. And then finally that gets inputted into the database or computer there, and then labeled for yes. Return back to the lab.
Very cool. And look at those weird creatures. I love it.
Yeah. And then there's a view of all the freezers that we have that are probably, some are fridges, but a lot of them are freezers and they're going to be full soon.
Any that we can peek into real quick?
Let's see.
That one.
The left freezer. Okay.
This is the right freezer. Oh, well.
Oh yeah, so these are one specimen there, but lots of ice packs that we use to keep things cold.
We're gonna just put that back in. Yes.
Yeah. Back here we have stations that are like designated for dissections of invertebrates and fishes.
Yeah.
And then also there's also like saving of animals too for preservation for museum archives and stuff that kind of undergo around here.
Super cool. This is so much fun. I just like the scale of this is fantastic. I hope you're having the best expedition. Certainly so good to get to see you guys out in the field. I know we're doing another one of these in just a few weeks. So if any of our audience wants to follow along with that, outstanding. Before we wrap up, we're at the 40-minute mark and time flies and you're having fun. Is there any final message you want to leave us with about this expedition, about how kids can follow along with more? Anything before we say goodbye?
I think, oh, go ahead.
Oh, no, I was just going to say thank you so much for being curious and like tuning in. It's super exciting to like do this science, but also it's more important to like share it. And it's so cool to have so many people engaged in what we do. It's really fun and cool stuff, and everybody here is great to work with. So I'm super excited that there's like interest in what we're doing, even when it seems like kind of silly that we're looking at poop, and I'm glad there's interest in that.
all of science is looking at poop. Everything in science is just ultimately poop based when you get down to it.
We all turn to poop.
What were you going to say? Sorry to interrupt.
Oh, no, all good. I was just going to kind of second what Sydney was saying, and like, your curiosity is a good thing, and it's a joy to be doing this work, and we're very grateful to be here and piecing together all of these amazing types of samples and all of this science that's going on all at once. It's incredible, and it's great to be a part of.
The video that I'm showing you just now is a video that Sydney sent me of the copepod sample from the bucket.
Oh yes, the mesto. Yeah, I have these fun little incubations that size fraction different zooplankton that we're interested in, and at the very bottom of it, it kind of keeps their poops after roughly a 12-hour incubation. So it's a nice like separation of that, and you can see that those amphipods there are super excited, and it's hard to see, but at the bottom you do see some beautiful specks of poop. Fecal pellets.
The prettiest poop I've ever seen in my life, and thank you very much for sharing. I want to ask one more final question before we say goodbye, but just a great one to end with, especially from our grade 11 class. What did you do in school to get this awesome job? Like, have you always wanted to do this? Is there a path that our students should take to sort of follow in your footsteps and end up having gourmet 5-star chef stuff while looking at poo all day? Like, what do we do?
Yeah, my path wasn't too linear. Like, I started off doing research in like ponds and lakes. I'm actually from Ontario, Canada. So I started off doing more freshwater stuff, and then I then moved on to like more estuaries. So this more oceanographic, like offshore research is kind of newer to me. But I think I got really interested in science because I grew up going fishing outdoors with my parents. So I always like appreciated being outdoors. And then through an internship with Fisheries and Oceans Canada, I was able to become like a fisheries technician in the summers. And I was like, wow, you can be paid to like measure fish. This is awesome. So I kept on like pursuing that route for me. Yeah.
What did you study at school?
Yeah, so I started off with like biology in my undergrad, then moved on to my master's, which was like ecology and evolutionary biology, and then my PhD was in marine biology.
Awesome. I am originally from Durango, Colorado, and wanted to go into emergency medicine. And when I went to college, I decided to do research to bolster like my experiences within science, and ended up doing a lot of research in biological oceanography, so doing genomics and transcriptomics on small marine heterotrophic bacteria. And during college, I studied molecular and cellular biology and biochemistry, and then decided that I wanted to continue doing research and going on research expeditions. And so now I'm in my PhD to do that.
How cool is that? Well, thank you both for sharing that. I've got to say, as cool as Colorado is, as a fellow Ontarian who has a degree in ecology and evolutionary biology, I've got to give you, I'm a little biased, I got to say. But thank you both so much for taking us on the journey around the ship. We can't wait for our next broadcast with you, and a huge thank you to our audience from all across the continent and beyond for joining today. But hopefully it goes super, super well, and you collect the coolest, weirdest specimens of things that we've never found in the history of man tomorrow or today. And till then, have a wonderful rest of your expedition. Thanks, you two.
Thank you.
Thank you so much, Jesse.
Thanks for having us.
Farewell, guys. See y'all soon. Farewell to our classes. We are